So the stages of the cell cycle, and the proteins that control it are similar in all eukaryotes. The stages of the cell cycle and the proteins that control them and the proteins that control it are similar in all eukaryotes. So in all eukaryotes, they are similar. For example, scientists have demonstrated that some of the molecules that regulate the checkpoints in the yeast cells can work in human cells too. So such similarities suggest that eukaryotes share a common ancestor. So you should note that such similarities suggest that eukaryotes share a common ancestor. That eukaryotes share a common ancestor. Or common ancestry. So this shows that they share a common ancestry. So what do you think would happen if, for example, the checkpoint stopped working in cells? Actually, this would lead to a disaster for the cells. Maybe it would lead to something like cancer and such stuff.
Mitosis. In this module, you will learn about mitosis.
So we will be looking at actually what mitosis is. So you can see here, to tell what mitosis is, you can see that mitosis is the division of the cell nucleus and its contents. So we are having a single cell here. It is now the parent cell, or the parent cell. So the parent cell undergoes mitosis. We are having now two nucleus. Then it will go through, so here nucleus divide, so we have two nucleus. Then the cytoplasm divides, cytokinesis, and we end up with two daughter cells. So cytokinesis divides the cell cytoplasm. Cytokinesis divides the cell cytoplasm. Mitosis as a process is very essential in, for example, regeneration of body parts. For example, regeneration of body parts like in lizards. But it's also responsible for growth and division. So if you look at it, we are having here two daughter nuclei. So at first it was single, then we have two nucleus, and that is what we are calling division. At the end, the phases of mitosis include the prophase, metaphase, anaphase, and telophase, which are 4 phases. And then the last one is cytokinesis, where a cleavage furrow will happen and the cell will divide into 2. That is in eukaryotic cells. During interphase, you can see that the cell is preparing to divide. So we have 2 centrioles. Then it will enter into prophase. During prophase, the chromosomes now, the DNA, will tangle and form chromosomes. So each chromosome is joined at the middle, Each of it is joined at the middle, as you can see. So this one is a chromatid, this is a chromatid, and it's joined here by the centromere. So when it condenses to form chromosomes, these chromosomes enter another phase, which is metaphase. So they will align here at the center. That is, during metaphase, they will align themselves at the center of the cell, and this is how they look like. So equatorial plate, they will align themselves there. Then the sister chromatids will be pulled apart by the spindle fibers and to the poles. So we have kinetochore holding it, and this will be pulled to the poles. And this is now another phase, which is anaphase. So sister chromatids will break at the kinetochore, and they will be pulled to the opposite pole, and thus dividing the nucleus into 2. The nuclear membrane, which are dissolved, will come back, and now we will have telophase. They have been pulled to the poles. They will tangle again and recoil and form new nuclear membrane. Then we will go through cytokinesis, the point where now the cytoplasm will divide. So we have cleavage furrow in animals, but in plants that is a different case. In plants, because they have cell wall, they will develop a cell plate before division takes place. So after the development of the cell plate, then the 2 daughter cells will form in plants. So after seeing the video, let's discuss the details little by little. So what might happen if the G2 checkpoint stopped working in cells? So if the G2 stopped working in cells, then this would be a disaster for the organism, because the cells that will develop might not be actually may not be prepared for division, and they may end up bringing or causing deformities and other diseases, or they may end up even causing the worst of calamities, and that is cancer. Cells divide at different rates. Cells divide at different rates, and that is what we are going to look at now. We are going to see that cells actually divide at different rates. Not all cells will divide at the same rate. So depending on the type of cell and the work that it does, these are the things that will determine whether a cell divides at a fast rate or a slow rate. So the prokaryotic cell cycle is similar but not identical to that of eukaryotic cells. So you have to recall that prokaryotes do not have the membrane-bound organelles and cytoskeleton found in eukaryotes. Thus, prokaryotic cells typically divide much faster than do eukaryotic cells. Prokaryotic cells divide pretty faster, divide much faster than do eukaryotic cells. So they will actually divide faster than the eukaryotes. So the rate at which your cell divides is linked to your body needs for those cells. In human cells, for example, the S, the G2, and the M stages together usually take about 12 hours. In humans, in human cells, it takes the S, the G2, and M stages together. So all these three stages together usually take about 12 hours. Usually take about 12 hours. So you can see that if you are studying cell division, you have to be very patient. So the length of G1 stages differs most from cell type to cell type. But you should note the length of G1 stage the length of G1 stage differs most from cell type to cell type. From cell type to cell type. It will differ from cell type to cell type. The rate of cell division is greater, of course, in embryos and children than in adults. Their cells cycle is also shorter, and many of their organs because many of their organs are still developing. But the rate of cell division also varies with different tissues of the adult body. For example, the internal lining of the digestive tract receives a lot of wear and tear. Because it receives a lot of wear and tear, it will be very necessary for it to be replaced as fast as possible. So in contrast, cells that, for example, make up the rest of your intestine, that is the smooth muscles, and many other internal organs, such as lungs, kidney, and liver, divide only occasionally in response to injury or cell death. So unless the cell dies or there is an injury, that is when there is a need for them to divide again. So cells that divide only rarely are thought to enter a stage that some scientists call G0. Cells that divide only rarely are thought to have entered a G0 stage. So you can see that they have entered another stage, and we are calling this stage the G0. So we will stop there for now. The next video we'll be looking at the details of this.